The hydrological restoration carried out by the Comprehensive Everglades Restoration Plan, also known as CERP, is important because seasonal fluctuation of water depth and flow velocity maintain ridge and slough composition, which structures the food webs within the Everglades. To evaluate the effectiveness of restoration programs, our lab conducts field studies to collect environmental data to assess biomass and species diversity estimates of wetland habitats. Although we have many sample sites spread across the greater Everglades ecosystem, sampling within each site is allocated to areas that are easily accessible by airboats, helicopters, and allow for the use of throw trap sampling. Because of the wetland habitat structure, only 50% of the sites are deemed sampleable, which led us to ask the question, will our estimations in aquatic animal and parasite be significantly different when considering habitat stratifications across our sample sites? So to investigate this question, we generated vegetation maps for our 146 sample sites using a random forest classification algorithm and multispectral satellite data. So far, we found across our study area that 55% of the identified vegetation was sampleable, 35% was non-sampleable, and 10% was non-aquatic habitat. From the results, we are currently working on finalizing estimations for scaling up surveyed biomass and density data to the regional scale using the weighted probability for each sample site based on the size of sampleable habitat within sample regions. We believe the results from this study will help us to establish an understanding of the relationship between habitat diversity and fish communities and to improve the usefulness of monitoring programs of the Florida Everglades. Thanks for listening and stay tuned for more updates on this project.